Okay, so this is a newer topic for AP Bio since the revision in the beginning of 2025 right now. And so this topic is AP Bio topic 2.6, and it is going to specifically focus on facilitated diffusion. Now, we've already seen some of this. If you are following along in my Unit 2 playlist, then we've talked about some of these concepts before, and then we're going to add to it at the end of this video. So right now, based on what we've already learned in Unit 2, what chemical properties would exist for these blue and green solutes or molecules on the screen, right? So hopefully you're thinking that these are probably ions or polar molecules. And so when we talk about facilitated diffusion, it is a type of passive transport, so there's no energy required in facilitated diffusion. And the molecules are moving from high to low concentration down their concentration gradients. So one example we saw, I think it was in maybe 2.4 or 2.5, is the example of water. So, I think it was 2.4. But anyway, so here water is a polar molecule. And when we talk about how molecules cross a membrane, polar molecules are usually repelled from those fatty acid tails. And so we have in our cell membranes specialized protein channels called aquaporins. So aquaporins are special protein channels for the bulk transport of water. Now, keep in mind, some water molecules, they are small and polar. They can go directly through the membrane, but the majority of water is going to travel through aquaporins. Now here I have all the water moving in one direction, but water can go back in the other direction as well. It depends on solute concentration and osmosis, which is the topic of 2.7, which is the next video in this series. So to summarize, aquaporins transport large quantities of water across membranes. Now, another example of facilitated diffusion and the use of a protein channel or a carrier protein to move large polar molecules across the membrane would be the case of glucose. So glucose is in our blood sugar. We use it for energy in cellular respiration. We store it in our liver. We store it in our muscles. So we are very dependent on glucose in our bodies. So when glucose is in high concentration, maybe outside of our cell, maybe you just had a meal or something, and we're going to move glucose into the cell. So glucose is large and polar, and it actually, when it binds to a membrane protein, the protein undergoes what we call a conformational change in shape, which means the shape of the protein changes, and that polar glucose molecule is able to enter into the cell. Now, this requires no energy, even though the protein is changing shape. Maybe like a decade ago, I looked into how does this happen, and scientists still hadn't figured it out. But to be fair, I have not researched it in years since. So to summarize, there are specialized proteins for the movement of glucose. They're usually like GLUT transporters. When we get to Unit 4 and talk about insulin and blood sugar balance, we will be talking about the GLUT4 transport protein. Anyway, when glucose binds to these proteins, they undergo a conformational change in shape, which allows the glucose to move down its concentration gradient from high concentration to low concentration. So this type of facilitated diffusion enables the movement of large polar molecules through membranes with no input of energy. Okay, so the next topic is we are totally going to switch gears, and this is something newer from College Board that I'm noticing that they are emphasizing in the new course framework, and so it is this discussion on polarized membranes. Now, in my opinion, to fully understand the whole big picture of polarized membranes and membrane potential, it would be helpful to learn it in the context of the nervous system. However, I am not going to spend a whole lot of time on nervous system, so I'm going to try to hit the key points of what a polarized membrane is and what membrane potential means and how it relates to facilitated diffusion. So, polarized membranes exist when there's an uneven distribution of ions across the membrane. So, when we think about ions across the membrane, remember they can only cross through a protein channel. And so if you have a fatty acid center of the phospholipid bilayer, that does create like a separation of ions. They cannot go by simple diffusion. So when you have this uneven distribution of ions across a membrane, that membrane becomes polarized. Now, this is not the case in all membranes. For example, red blood cells do not have polarized membranes. It is mainly important or characteristic of certain cells like muscle cells and neurons. Neurons are like your brain cells or your nerve cells, that for them to function, they rely on this polarized membrane and a difference of charge across it. So we're going to zoom in to a neuron's membrane. So you can see how that pink neuron on my screen has that really long axon, is what it's called. That is what we're going to go ahead and zoom into. So this pink neuron here on the screen is one cell. It's just a very highly specialized cell. So as we zoom in, we're going to say that the inside here between the two layers is the two lipid bilayers is the intracellular portion or the cytoplasm. Then we have outside the cell in white. Now, in that lipid bilayer, there are protein channels. We are talking about facilitated diffusion, so we're looking at the movement of ions across a membrane, but through a protein channel. Now, to understand polarized membranes, we need to add some background knowledge to what we are looking at. So inside of our cells, I want you to remember from Unit 1 that we have a lot of DNA. We have base pairs, we have nucleotides, and in a nucleus of one cell, there are 6 billion nucleotides. And each nucleotide has a phosphate group. Now, a phosphate group has a negative charge. Now, not only do we have DNA that has lots of phosphate groups, but we also have ATP. We have millions of ATP and ADP molecules within our cells, and these ATP molecules have 3 phosphate groups. Each phosphate group has a negative charge. So we have a lot of negatively charged molecules within the cell, in addition to negatively charged ions. I have chloride ions here, but there's going to be more variety as well. So we want to keep in mind that we have negatively charged molecules within the cell, even though as we continue, we are focusing on 2 different positively charged ions. So here on my screen, we see how there's lots of sodium outside of the cell and potassium inside the cell. Keeping in mind that inside the cell has lots of negatively charged phosphate groups, negatively charged amino acids, ions, etc. So when we look at the overall charge within a cell, I also want to point out that the membrane also has some specialized like proteins that allow potassium, a positively charged ion, to actually diffuse out of the cell through that open protein channel. Now, this allows the inside of the cell to have an overall negative charge. So the overall negative charge within the cell is negative in comparison to the outside, like relatively speaking. And so if you think about that potassium though, and you're like, well, why doesn't all of the potassium just diffuse out, right? There's a high concentration of potassium within the cell. Well, if you look outside, it's a positive charge, and positive repel each other. So there will be a limit to how much potassium actually leaks out of the cell, because then it would just be moving into an all positive environment. Okay, so now though, let's go ahead and define a few words. So I'm going to kind of cover up the picture a little bit. So this membrane is polarized. Earlier I said a polarized membrane exists when there is a difference of charge across the membrane, really due to different concentrations of ions. Now, at rest, if you compare the external to internal, you compare the charges across the membrane, the inside is more negative relative to the outside. If you were actually to measure this with like a voltage meter, it actually on the inside at rest with no action potentials or no activation, the inside would be negative 70 millivolts in a nerve cell at rest. So in general, when we talk about polarized membranes and we talk about membrane potential is the electrical potential difference across a membrane. So here it would be negative 70 millivolts. Okay, so let's go ahead though and see what happens. Now, this is in an axon of a neuron. So I'm not going into detail what happens at the dendrites or the top or the front part of a neuron. When it receives a message and that message is using neurotransmitters, there are changes in ion concentrations that flow into or out of this neuron. And so let's pretend a whole bunch of positively charged ions flow in to the neuron. That's going to change the voltage within the cell. Okay, so these purple and green proteins on my screen are actually voltage-gated ion channels. When the voltage or the charge inside the cell changes, that triggers the opening of this gate, this voltage-gated ion channel. Now, this only happens if the neuron receives a signal or a message, usually from a neurotransmitter. Okay, so once that voltage-gated ion channel opens, now sodium ions can rush inside the cell. I only had a few go, but you can imagine they would rush inside. Being positively charged, they're going to rush inside down their electrochemical gradient to where it's more negative inside. Now, this sodium gate does not stay open. Eventually, I forgot to mention, if we notice, we just had a whole bunch of positively charged ions flow into the cell. So now there's a reversal of charge. This is actually called depolarization, where the inside is more positive compared to the outside. So the purple sodium voltage-gated ion channel actually will close, and then the potassium voltage-gated ion channel, now that the inside is positive, that triggers the opening of the potassium voltage-gated ion channel. And all that positive and positive are going to repel each other. So given a chance, the potassium is going to flow out of the neuron, right? Flow out into the extracellular space. And over time, we'll have a reversal. We'll have mostly potassium on the outside and sodium on the inside. Now, this potassium voltage-gated ion channel will close. And at this point, both gates are closed. However, the ions are in the opposite locations. We have potassium on the outside and sodium on the inside. So, what has happened so far is related to facilitated diffusion. Facilitated diffusion is going to be the diffusion from high to low concentration or down the electrochemical gradient through protein channels. This required no ATP. However, to reset this membrane does require active transport. So I'm going to go over it now in this video because it relates to what I'm teaching. And then also in topic 2.8, we will revisit this again. This is called, we need to basically reset the membrane. So we're going to use what's called a sodium-potassium pump. And there's that word pump. That implies it's going to be active transport. So here's how it works. We're going to require ATP, which is our energy currency of the cell. And you'll notice when that ATP attaches to the sodium-potassium pump, 3 sodium ions fill in or attach to the inside of that protein. Now, phosphorus is used, or phosphate groups are used a lot to activate or deactivate proteins. Okay, I'm going to slow down here. That phosphate group has a negative charge. When it binds to a protein, that protein is made of amino acids with different R groups. Those R groups have chemical properties. You add in a negative charge, that's going to cause some shifting in how those amino acids are bonded to each other and how they are interacting. And that's going to lead to, if you watch my screen here, a conformational change in shape. When that phosphate group attaches, it causes the sodium-potassium pump to change shape. Now, this sodium is able to flow out of the cell into the extracellular fluid, and 2 potassiums flow in. Now, once that phosphate group detaches from the sodium-potassium pump, the protein goes back to its original conformation, and now the 2 potassium ions are out. So, to summarize so far, when one action of a sodium-potassium pump requires an ATP molecule, we pump 3 sodium out and 2 potassium in. And this is why, another reason why, the outside is more positive because 3 are going out while only 2 were coming in, contributing to the overall return to negativity, a negative voltage on the inside of the cell. Now, I only modeled the sodium-potassium pump once, but over time, after resetting the neuron, we'll have an establishment of a high sodium concentration back outside of the cell. Potassium will be high inside the cell, and the charges will be reset with an overall positive charge outside and an overall negative charge inside. Now, as you're watching this video, you're hopefully thinking a lot. Sometimes when you study for a long time or you take a long test, you feel very tired afterwards. And that's because as your neurons are firing, this is happening and it requires energy to keep resetting your brain. You use a lot of energy while you think in this process with the sodium-potassium pumps so your neurons can keep firing. But anyway, let's go ahead and summarize what we've learned with this example of the neurons and polarized membranes. So one key thing I needed to teach you was that membranes may become polarized by the movement of ions across a membrane. The second thing we needed to see was that charged ions, including sodium and potassium, require channel proteins to move through the membrane. Now, the sodium-potassium pump was extra in this topic 2.6, but again, it'll show up in topic 2.8 as we focus more on active transport in that video. All right, great job, and I hope this was helpful.